The beginning of DNA replication begins with the double-stranded molecule unzipped into two separate strands. The four bases of DNA are thymine, adenine, cytosine, and guanine. To be able to begin the replication process, the base pairs must be broken, which is the job of helicase, shown as the orange Play-Doh. DNA helicase breaks the hydrogen bonds between base pairs, which separates the strands into the replication fork. The strand's replication order and process is determined by the significance of 5' and 3'. The leading strand is oriented in the 3' to 5' direction, while the lagging strand is oriented in the 5' to 3' inclination. The primer then binds to the 3' end of the strand as an introduction for replication, and the primers are generated by DNA primase. Enzymes known as DNA polymerase create the new strand via elongation. Polymerase III is the main replication enzyme, while polymerase I is responsible for air checking as well as repair. DNA polymerase binds to the strand at the primer site and begins to add new base pairs complementary to the strand when in replication. The lagging strand begins its replication with multiple primers. Each individual primer is several bases apart. The DNA polymerase then adds pieces of DNA called Okazaki fragments to the strand. This process is discontinuous as newly created fragments are disjointed. Exonuclease then removes all RNA primers from the original strands, proofreading the newly formed DNA for errors. DNA ligase joins the Okazaki fragments together to form a singular unified strand. Telomerase catalyzes the synthesis of the telomere sequence at the ends of DNAs. Now completed, the DNA strands coil into a double helix shape with the result of two DNA molecules, each with a strand from the parent molecule and one new strand.